Okay, wait a second. Okay. Okay, so today, this, as I just mentioned, this is going to be the beginning part of chapter 15. And this is what we would call a specific response. That's hard to say. Right? You'll also see it called the immune response. And your book titles it, I think, acquired immunity or acquired response, because it's something that your body has to actually do in order for it to work. Right? So I think. Sorry, this is actually chapter 16. Come on, you guys have to help me. I don't do this, so I put the wrong chapter numbers on here. Yeah, adaptive immunity. Sorry, not acquired. So it's 16. Adaptive. Some of the shadows. Okay. So this part of the immune response, unlike the non-specific defenses or the innate defenses that we talked about before, this is different for each individual bacteria and it's different for each individual virus. Your body does all the things we're going to talk about over and over and over again for each one. Okay. Traditionally, we kind of divide this into two branches or arms or areas because it's really difficult to explain it to you in real time with both of the branches working together. So we're going to split it up into what we traditionally call the humoral response. And I'll just put a box around that to make it look more interesting. And the cell-mediated response. And right now, what I'm going to do is go over like a kind of an overview, a general, this is where we're going. And then the rest of today, we're going to spend on the details of the humoral response. And when I see you again, we'll do the cell-mediated response. Okay. So the humoral response is all about producing some cells that are going to make immunoglobulin as their final product. But it actually starts, both of these start with cells that are going to come from the bone marrow, like we talked about last time, right? Those totipotent or hematopoietic stem cells. And the fetal liver actually is also involved in the generation of these early cells. From the bone marrow, as far on the humoral response side, the cells that come out of the bone marrow, we refer to those as pre-B cells or immature B cells. Because they're not ready to fight an infection yet, right? But how we know what they are and how we can find them in the blood, for example, we know that they exist because I'm going to draw them in purple and give them a nucleus, right? Remember, they're a lymphocyte, so they're part of the white blood cells. And on their surface, every one of them has a receptor that is an immunoglobulin, and it's actually immunoglobulin D. So I'm going to write off to the side here, IG, capital I, lowercase g, capital D. Immunoglobulin D. It has it on its surface. And it's going to function as a receptor to bind to a bacteria, a virus, a protein. And another marker that these immature B cells have is something called CD28. They're CD28, no, it doesn't look like an 8. CD28 positive. So those two things, when cells have those two things and they're floating around in your bloodstream or your lymph, your body's like, oh, that's an immature B cell, right? So that's where they end up. From the bone marrow, they're going to go into lymph. And from the lymph, they're going to go into a lymph node, or maybe to the spleen. And they're kind of cruising around and they're waiting to encounter antigen. Because once they encounter antigen, that's what's going to trigger them to become mature. All right, so we need to digress for a minute and talk about what is an antigen. So I'm going to put that over here a little further away. Leave some space because I'm going to keep going this way with this. So just leave a little space, either sideways or underneath, wherever you are in your notes. So we abbreviate antigen, capital A, lowercase g, like the element, right? Like gold, even though it's not. And what an antigen is, it is a non-self molecule. We used to say a foreign molecule, meaning not part of me. Try not to just think about the humans and think about ourselves, right? So if I'm a goat and someone puts sheep's blood into me, that is not me. And that would be an antigen, right? Just like for you, pollen might be an antigen, or horse serum might be an antigen. Why would I get horse serum? Because that's where they used to get insulin from before they could make it DNA and recombinant. Okay, the best antigens are proteins. Yay, great antigens. Plus, plus, plus. Sometimes peptides, if they're long enough. And as I mentioned, things from plants, plant products, your book says. Okay, that means pollen. I don't know why we can't just write pollen. Right? Blood products. And then, of course, things from bacteria, viruses, our friends, the fungi, parasitic worms, right? Polysaccharides, carbohydrates, remember that from BIO1, right? Polysaccharides can be antigens, but they need to be gigantic and they're relatively poor. But if you take a polysaccharide and you attach it to a peptide, right, that makes it a better antigen. But this kind of helps if you, if we just speak about vaccines for just a minute, vaccines like tetanus toxoid, because that's a protein, we can inactivate it and it's a protein, that's going to be a good antigen for you to make an immune response to. But the bacteria, Streptococcus pneumonia, remember that one? We met that one when we talked about Griffith and his experiments and the dead mice, right? Streptococcus pneumonia is covered in a capsule. And a capsule is mostly sugar. It's mostly polysaccharide and not as much protein. And it's 23 different sugars. So it took a really long time to make a vaccine against pneumonia. There is one now, right? There's one for infants and there's one for old people, right? They call it multivalent, right? But that's why, well, like when I was in graduate school in the Stone Age, people were like, oh, we should make a vaccine against pneumonia, against Streptococcus. And everyone was like, it can't be done, right? It's all sugar, it's all polysaccharide. Eventually, right, science moves on and we can do hard things. All right, so when I say that the pre or immature B cell has to encounter antigen in order for it to get triggered chemical messages for it to become a grown-up B cell, right? We refer to this as B cell proliferation, or actually we call it clonal selection. because only the B cell, only the pre-B cell that matches the particular antigen is going to be triggered to proliferate and differentiate. Can I erase the part about the antigen because I need more space over there? Yes, you guys okay? So only the immature B cells with a matching IgD receptor, right? So the IgD binds to the antigen. Those are the only ones that are going to be selected. They're the ones that are going to mature. So I'll redraw my B cell here, my immature B cell. I'll make them a little smaller this time. And I'm trying to draw an IgD that has, that looks like it has a triangle receptor, because I can draw a triangle, circle, square on a good day, maybe a diamond. So this IgD is, this immature B cell is specific for triangle antigen. You have other immature B cells that might be specific for circle, half circle antigens, right? Like that. You may have other B cells that are specific for square antigen. Remember, I told you those are the only ones I can draw, right? You have 23 million combinations of receptors, so much more than triangle, circle, square. And your body makes those based on the genes that you have, because those are genes you carry because your ancestors survived a particular infection, right? 23 million combinations before you've ever met an antigen you can make. Right, it's just like big mind blowing. Okay, so we're gonna trigger triangle antigen. So I'm gonna draw our classic friend that I'm always drawing on the board, and I'm gonna put triangles on the surface of this bacteria that kind of looks like E. coli, right? So that you can imagine triangle antigen binding to IgD with triangle shaped receptor, right? Okay, when that happens, when antigen from a bacteria binds to this particular, it's going to bind to and trigger this particular immature B cell to proliferate and differentiate, not these guys, because this one's specific for squares and this one's specific for circles. So maybe this one's specific for, you know, hepatitis virus protein of some sort, and this one's specific for tetanus toxoid protein. Right? This B cell is going to first proliferate, which for me, what does that mean, proliferate? Reproduce, what do we call that in eukaryotic cells? Mitosis, very good. Right. Because we want more of these B cells now. We want ones that are going to be able to fight this infection that's in front of us right now in this lymph node, right? We don't need to get ready for a hepatitis infection right now because we don't have one, right? Or a tetanus infection. So this immature B cell is going to proliferate, and I don't know, I'll make like 4 of them. Because we need lots of these that are all specific for triangle antigen now. Right? And they're going to go through several rounds of mitosis. I'll have this guy do some more mitosis, and this guy. Then they are going to differentiate, meaning they're going to change into cells that function differently than this cell would. And the cells they're going to differentiate into are, some of them are going to become what we refer to as B memory cells. or memory B cells. People say it both ways, right? So I'll make these guys up here be some memory B cells. They're adult now, they're ready to go, they're fully mature. And what's the difference between the immature version and the mature version? They're a lot closer to being able to be differentiated into the functional cells, which are the plasma cells. And we're going to keep these for later. They're going to live in the lymph node where this first immature B cell saw the antigen. They're going to live in that lymph node or in the spleen. And they might travel around part of your body, but they're always going to go back to that lymph node where they saw antigen first. And they're going to hang out there, sometimes for the rest of your life, sometimes maybe just for a few months, right?
Are B cells leukocytes?
Yes. Yes. So they're part of the lymphocyte side, right? Yep. And we would call them leukocytes because they belong to that group, large group of white blood cells, right? But they're not granulocytes, right? So those other ones we talked about, basophils, mast cells, eosinophils, those are also leukocytes. So usually we refer to the B cells as lymphocytes, but people do use those interchangeably. So these are for later. They're going to live, like I said, in your lymph node. They're going to travel around the lymph vessels. And the rest of these guys are going to differentiate into a different cell that we call a plasma cell. And a plasma cell, it looks different. It's a different shape. It is a cell that is like filled with endoplasmic reticulum and Golgi and mitochondria, because these plasma cells, they are protein factories. And the protein that they're making is immunoglobulin. So I'll put a nucleus there. So these guys are going to become plasma cells, and they are going to produce immunoglobulins that are all specific for triangle antigen. And they are going to, so I'm going to put immunoglobulin, which we abbreviate capital I, lowercase g, right? That's the same thing as saying antibody, or antibodies, but that's kind of an old-fashioned word. People don't use that as much anymore. You won't see that as much in your book, right? These immunoglobulins then are going to get dumped primarily into bloodstream. It's supposed to be blood, so that they can go around and move from the blood to the lymph and the lymph back to the blood, and they can find other triangle antigens and bind to them. Okay. So the major effector molecule here that is made by the plasma cell, that is differentiated from the B cell that came from the immature B cell that came out of your bone marrow, right? All that long time ago. The main product, the main protein product that's made is immunoglobulin or antibodies. So I'll just put some more of them in here, floating around in your blood, like that. And I'll put IG, immunoglobulin. Now, so what? What's the point of immunoglobulin? We'll talk about that in a minute, okay? I want to go back to this for briefly, the cell-mediated response. I'm going to put it on top of and below just to remind you of the rest of the other side of the, like, the overarching picture. The cell-mediated response is all about making a different kind of lymphocyte that we call a T cell, because they go to the thymus. That's what they're called, thymocytes. And we just abbreviate them by calling them T cells. And the thymocytes are going to differentiate into basically 3 different types of cells. Okay. They can become what we refer to as a T helper cell. So capital T with a subscript of a capital H. And as you might suspect, they help. What do they help? They help other T cells, and they help these B cells become mature. Okay. They release a set of cytokines that we specifically refer to as interleukins because they are chemicals that leukocytes use to talk to each other, or lymphocytes use to talk to each other, right? So their job is going to be to produce cytokines, particularly the ones we call interleukins. And just as a little warning, those are going to be abbreviated capital I, capital L with a dash, and then a number, like IL-1, IL-7, IL-4, IL-18, right? Interleukins, because they're communication between the leukocytes. Some of them are transcription factors. Or they're going to develop into cytotoxic T cells, which we abbreviate capital T with a superscript of a capital C. Cytotoxic, and that's what they do. They kill infected cells. Okay. And the third type, we call those T regulatory, or sometimes people just call them T-reg, and that is pretty self-explanatory, right? Their job is to help kind of contain the immune response. Their job is to dampen it down at times so that there isn't too much inflammation or too much of the end products of turning this whole system on, right? So this B cell response that I showed you here, this humoral response of the pre-B cell going out to the lymph, meeting up with antigen, some of the pre-B cells being clonally selected. This is the winner this time, right? And then proliferating and differentiating. In the example that I showed you across here, we would call this a T-independent response. Because, right, I didn't show you the involvement of any T cells or any of their cytokines, any of those interleukins that come from the T cells, right? So this clonal expansion, in this particular instance, the way that I just showed you guys, if I put a little orange asterisk here, this is a T-independent antigen. Or you'll hear people say a T-independent response. Antigen response. Anyway, I'll just add more words to it. Does this help you if you're sick? Absolutely. But it's thousands of times better if you get help from the T cells. It's faster. You get more clonal expansion. You get more mitosis. You get more plasma cells made if the T cells help the response, right? If you are immunodeficient and you don't make enough T cells, right? Can you see how you'll be able to fight some infections, but you might not be great and you might feel like you're sick all the time because you don't have a T cell helping response. That is one of the major reasons that people get so sick when they have human immunodeficiency virus, HIV, is because that's what the virus kills. It kills your T cells. And when you don't have T cells or your T cell number goes down dramatically, you can't fight off anything else. You can't fight off a fungal infection. You can't fight off a viral infection from a different virus because you lose the help. Okay. So next time we'll go through how it helps, blah, 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 right? So just hold on to that for later. Questions about any of this before I go on?
Did you go over interleukins one more time?
Barely, right? So they're chemicals that are produced. They're cytokines. Technically, they fall under the category of cytokines, but cytokines include everything like histamine, which causes kind of generic responses, and prostaglandin, which causes kind of a generic response. Those are all considered cytokines, right? Interleukins are much more specific. What that means is usually the sending cell produces only a few interleukins, and the cell that it's sending that message to has a perfect receptor for that particular message for it to do something else very specific. right? So it's for communication between the leukocytes so that they become better effector cells, right? To help or make it worse. We originally named them, why do I say we? Because this is what I did. This is my PhD is in immunology, right? 100 years ago, right? There weren't as many interleukins then because we named these molecules, these proteins that T cells made and B cells made and mast cells made, and macrophages, we named them by their function, right? We would say, oh, this macrophage makes this molecule that causes or helps B cells. So they called it that. B cell growth and differentiation factor, right? Great. And that was helpful in a way until you just only saw the letters. It was helpful in a way because you're like, okay, I know what the function of this molecule is. But then when they found out that same molecule also stimulated cytotoxic T cells and they had to add on more letters, it started to get ridiculous. And so they were all renamed, not by their function, but just with the numbers, because it turned out that a lot of them did more than one thing. I think we're up to interleukin 21, 23-ish now. When I was in graduate school, I think we were on 12, right? But that was like 30 years ago. So we're still finding them because this is a super complicated, very small, short-lived molecule process. You know, this molecule might be there for a couple hours and then disappear. All right, so I'm going to erase this. Maybe it's a pretty nice picture. Yeah, I'm going to erase it. And I want to talk to you a little bit about next. Let's check my notes here. Oh, yes. Okay. So now there are some, staying on the topic of antigens. We're going back to the topic of antigens. I should have done that first. So we talked about non-self antigens, right? Like things, proteins on bacteria and viruses that can trigger a B cell response or trigger a T cell response. They actually start, we could say, the adaptive immune response. And as we said, they're mostly proteins, right? But just to make it extra confusing, you also have some antigens that belong to you that help your immune system cells know that this liver, this stomach, this heart belongs to Gail Stewart, not to anybody else. Okay? We call those self-antigens. They are also proteins. They are going to be on the surface of your cells. And I'm going to hyper underline your cells because they're unique to all of us, right? The more closely related we are, the more likely we are to share these antigens, right? The further apart we are, the less likely we are to share them.
Is that why some people that use organ?
Yes, this is totally what organ graft versus host and host versus graft is about, right? So these self-antigens come in groups. There are 3 of them. We're only going to talk about 2 of them. We call one of the groups, well, all of them are called histocompatibility antigens. Right, that's where they were discovered. Cells being compatible. If I take a skin graft from this mouse and put it on a different species of mouse or a genetically different mouse, will it take or will it not take? That's where they were first discovered. And sort of some of the features worked out. So we call them the major histocompatibility complex. But that's a lot of writing, so we abbreviate that as capital M, capital H, capital C. No, it's like another language, the immune system. Right? It's like we have to make up some new words because we don't have enough science words. Right? In humans, we call it the MHC in mice and rodents and bunnies, but in humans we call it the human leukocyte antigens, HLA. But most of the time, people just call it the MHC because that's what we're all more familiar with using, right? But in humans, you might see it called the HLA. That's kind of like, you know, when we say it's called the Krebs cycle and the citric acid cycle and the TCA cycle. It's kind of like that. I'm going to use MHC because I worked in mice. So now, class 1, Roman numeral 1, MHC. This is a protein that is unique to you and is present on all your nucleated cells. So that means everybody, all your cells have it except your red blood cells, right? And apparently it looks kind of like this. I'm going to draw, like the book did, I'm going to draw phospholipid bilayer here. And I'm going to draw a class 1 MHC sticking out of it. And it kind of looks like this. It has a tail and a ball here and one there and one there. And then a yellow thing that sticks out this way. Okay. That's class 1 MHC. So mine is unique to me. Yours is unique to you. It's on every single one of my cells except my blood cells, my red blood cells. And that is literally how my immune system, when my leukocytes and my dendritic cells, when they're laying around and traveling around, that's how they know, oh, this is self. This is me. This is me. Okay, this is me. This is not non-self bacteria or foreign virus. This is me, right? Don't attack that. That belongs to us, right? When someone has an autoimmune disease, that's when some of this is broken down and your immune system cells don't recognize self, right? Okay, so that there. You also have on the surface of your cells of the immune system, you have class 2 MHC. So this is on the surface of, I'm trying to look and see what it actually says in your book. It just says on some types of white blood cells, some types of white blood cells. So white blood cells. Like a B cell or a T cell or a macrophage. And so these cells still have class 1 on them to tell the body, hey, this is me. But they also have an additional molecule, this class 2 MHC. So I'll give a little space here. The surface of your cells have so many receptors on them, most of which we barely understand what their function is. So this one kind of looks like this. So some through the membrane, and then it looks like this, and like this, kind of like kidney shaped, kidney bean shaped. So this is class 2, right? And here's the class 1, because it's present on all my cells, so that my body doesn't mistake a macrophage for a foreign cell. And what I want you to, in particular, to notice about the class 2 MHC is here, this area, and it can be different shaped. That area is to bind antigen. We call that the antigen binding site. It's usually not the whole bacteria this time. I'm just gonna, should have only lined it red. It's not going to be the whole organism this time, but probably just a piece of the organism. So it's a little different than how that IgD bound to the bacteria, but it's a place where antigen can fit. That's going to be on the surface of T cells, on the surface of B cells, on the surface of macrophages, on some dendritic cells, sometimes a natural killer. So on different kinds of cells, we're going to see both, some, one, plus there's a whole bunch of other nonsense on the surface of these cells too, especially the T cells. We'll talk about that, what the surface of the T cell looks like later, next time. Okay, so what I'd like to go back to now is I would like to talk to you guys a little bit about immunoglobulins. So I'm going to draw that probably. And then functions of immunoglobulin and the different classes of immunoglobulin, and then we'll go to lab. I know it's a little long today. It's a little bit time for me to set up, but most of you are going to be finished with your unknowns today. Yes, if you aren't already. So this is what you have to suffer through. We're going to finish this chapter, and then that's going to be it for the final. All right. So today, when I see you again next week, and then next Thursday will be a reading day for you. Okay. Today's Tuesday, right? No, today's Thursday. So when I see you on Tuesday, we'll finish this chapter. And then next Thursday will be a reading day, so you can study. And then Tuesday, the 13th, we will have our final exam. Okay? All right, so I'm going to erase this. Give you a little mental rest. Yeah. I know, of all the days not to have a black marker. Okay, oh, whatever. All right, so let's spend a little time talking about immunoglobulin. Okay, remember that's the final product made by the plasma cells. I'm going to draw a picture of like our poster child immunoglobulin, immunoglobulin G for gamma. Okay. And then when we look at the other types of immunoglobulin, you'll see some of the variations. Let's make it purple. So, protein, globular protein, immunoglobulin. That's right. You find it in the serum. All right, floating around and in the blood. So in your blood, serum. And it is 4 at minimum, it's 4 polypeptide chains. minimum. So I'm gonna draw IgG, which does happen to have 4. And I'll try and make it really large so you can see it. Okay, these 2 pieces, because they're the biggest, we call them heavy chain. H for heavy chain. And in an IgG molecule, there are 2 heavy chains. The heavy chains are held together to each other by disulfide bonds. So I'm going to put little sulfurs here, S's, indicating that those are held together right at that place where they bend. Most of these heavy chains, the part of the heavy chain, this is all the same. All the same amino acids in the same order. So this portion of the heavy chain, it's probably a little longer than that. This portion we call the constant region. So I'm going to put a little piece here and color it in like that and say constant region. Because it's the same. All the IgGs have the same constant region. This part, which I'm just going to put with little diagonal lines, that part we call the variable region, because that's the part that's different. Remember when I said you can make 23 million different combinations of immunoglobulin? That means 23 different million combinations of this heavy chain and the light chains I'm going to put next to it, right? And that's where the changes are in this variable region, not the whole thing, but just out at the edges. And then we also have 2 light chains in an IgG. And similarly, I'm going to put them here and make them kind of look like this. So that's light chain, L for light chain. And the same, the area that I've already colored in, that's the constant region of the light chain. And there are only 2 different versions of constant region. They are held to the heavy chain by sulfur bonds as well, disulfide bonds, also at the base of them like that. And then this is the open part, this is the variable part. Again, which is different. Oh, there we go. So my antigen, my foreign antigen, can fit in here. This is the antigen binding site. Right? That's where that shape comes from of triangle, circle, square that I was drawing on the IgDs, right? It's obviously a lot finer and super specific in a real immunoglobulin. That's pretty good. The only other thing that I need to tell you is, well, there's many things, but I'm going to put a couple bumps right here, like this on its side. Those are for complement fixing. Remember, I know it's been like over the weekend. No, it was only since Tuesday, so only 2 days ago. Remember we talked about complement? And we talked about how complement was an alternative pathway, a classical pathway, and a lectin-mannose pathway. And the classical pathway requires immunoglobulin, and this is how it gets turned on, because the immunoglobulins have a bump on them that can bind to complement and get it started. I just had like this horrible feeling. Did we not talk about that?
We did.
Tuesday. We did? Did we really?
You told us to remind about the neurons.
Oh, okay, we'll go back to that at the end. After I turn the video off. Okay. So that's what that's like the poster child of all immunoglobulins. What I do want to show you though on the when I share my screen with you is I have a picture of a real immunoglobulin, like 3-dimensional with a maybe there it is, this one. So let's look at this for a minute. Of course, my drawing is much better. Okay, now we've got nothing there. Of course, my drawing was much better. Yes, that's what you're all supposed to say. But this is what I was trying to illustrate. This is the little picture that I want you to see. That's the X-ray crystallography picture of what an immunoglobulin actually looks like, right? And they call that area here the hinge region because the antibodies literally can spin and twist and bend as needed. And the 2 antigen binding sites are also flexible, right? And can show up in any, this has been flattened so you can see, I think the green is the light chain and the blue is heavy chain, right? And then this would be, I think the antigen binding site is here in the middle, right? So you can kind of see what it looks like in real life rather than just how I drew it. Okay, so what can antibody do for you? This is the slide for that. Okay, I'm going to make a list off to the side while we're talking about this picture. And then hopefully I won't erase it before I stop sharing screens so that the people at home can see it. Right? So this is the outcomes, effects, right? Of immunoglobulin antigen binding. I like to call it what antibody can do for you, right? Like why do we care? Why do we need these antibodies or immunoglobulins, and what's their real function? And this is the picture. I'm going to jump around a little bit, but I'm going to start number 1 with this one here. One of the major effects of immunoglobulin binding to antigen is to do something that we call agglutination and sometimes precipitation. So you know what precipitation is, right? Droplets forming, particles forming, like in chemistry. Agglutination is to clump together. So if you think about it for a minute, and this illustration shows it pretty well here down at the bottom, right? Do you see how I've got, they look like IgGs, and I have all of the immunoglobulins are binding to 2 bacteria, right? And so now I'm clustering 10 bacteria, 30 bacteria all in one place. And that makes it easier then for the macrophages, for inflammation, for phagocytosis, for T cells to find them, all the things to actually kill the bacteria, right? So the immunoglobulins themselves don't actually kill the bacteria, but this is how they help with the killing and removing, right? So they clump them all together. Number 2, I'm gonna put these two together. Neutralizing and antitoxin. Neutralizing immunoglobulins, we usually think about those in terms of viruses because they're going to block the virus from entry into your cells, because what's going to happen is the immunoglobulin is going to bind to the virus before the virus can bind to you. Only if you've seen it before or if you've been vaccinated, right? Same with the antitoxin immunoglobulin. This is, oh, I've gotten a tetanus shot and now I stabbed myself with a sewing needle that I just picked up off the dirt because I thought it would be fun to sew outside because it's so nice and warm, right? And maybe I put some Clostridium tetani into my finger, but my body's going to go, oh, we know that one, right? So we've got antibodies for that, like in 10 minutes. Like for toxins, obviously, right? Like exotoxins. 3. I'm trying to make this list a little shorter than 6 things, right? Opsonization, and we may have mentioned that a little bit with complement. Opsonization is to coat. And so what can happen is immunoglobulins can literally cover the surface of a bacteria. And that has 2 effects. It inhibits the bacteria from doing its normal functions, like reproduction and metabolism and binary fission. It also makes it like a target for increased phagocytosis, because macrophages, let's say that's target for macrophages, because macrophages have a receptor for immunoglobulin heavy chain. So I know it's a lot to think about. So opsonization is to coat the bacteria, and that's going to do 2 things. It's going to prevent the bacteria from like importing glucose, right? So it's not going to be able to do its metabolism, and it is also going to prevent it from multiplying. And when you cover a bacteria with immunoglobulins, like here in this picture, the big purple thing is a macrophage. Macrophages have receptors specific for heavy chain of immunoglobulin. So when they run into that, it's going to be more effective for them to bring it in and kill that bacteria. And we have one more, number 4. We're almost there, guys. We have this slide and one more slide. I know it's a long lecture. The last one is immunoglobulin bound. Remember, oh, let's just say activate complement activation. Let's just say that. Complement activation, classical pathway. Classical path, right? So that's going to cause that membrane attack complex to form and holes to be pulled, made in the cell wall and cell membrane of the bacteria. All right, let's get rid of that. Just going to leave this for a second. So if you're watching it at home, you can put those words with this picture. And then pop that off for a second. Okay, questions about any of this? about what antibody can do for you before we talk about the different classes or categories of immunoglobulin. And then we can go to lab.
What is number 3? It says target for.
So opsonization, right? That's a target for a macrophage. That's the symbol for a macrophage.
Or it's gonna be like putting a red flag on it.
This is like putting a red flag on it, yes. That's like going, hey, macrophages, woo, come on over, feast time, right? This is more like smothering the bacteria so it can't do its metabolism and it can't get the things it needs to divide and the cell wall can't form right and causes all kinds of problems. Wait, and go on. Going to, I just can't erase it yet. Just have to let it sit there for another minute. All right. Okay, so now let's look at the 5 different types or classes, categories of immunoglobulin. And I will share my screen with you again and try to, cannot see anything. Oh my God. This, it's too many choices. There they are. Oh, woo, yay. Okay. Here are the 5 major classes of immunoglobulin. Now, there are more than this, but this is enough for us. Okay. I am not concerned about how much they weigh. I don't care, of course, but I am interested in the rest of these things. Okay. A lot of times people remember the letters of the immunoglobulins by the woman's name, Madge, M-A-D-G-E. That helps, right? We're just not going to do them in that order. Okay. But however, you can make up a song, draw them, list them, however you can remember them. So this is our friend IgG that we've been using as our poster child. And there's a reason that we're using IgG as the poster child. It's the reason that I drew it dumping into the bloodstream. And there's a reason I drew that one for showing you what an immunoglobulin looks like, right? Because it binds to antigens at a time, but it is 80% of the immunoglobulin in your serum at any given moment. So what that means is if I took blood from all of us right now and I put it in a centrifuge and I collected the serum and I did an immunological test to say how much of this is IgG for the whole class, of all the antibodies that I had in my pool, 80% plus would be IgG, right? It is the most prevalent. Look how long it lasts. This is a half-life. So this means this antibody was tagged radioactively, and this is how many days they could measure the radioactivity. It was 23 days. So that means it's a half-life. So that means it's actually around for 46 days, right? That's a month plus. So when you get when somebody in your house gets a cold, right? And like, let's say one of your children or your spouse or your roommate, and they're really sick and you get a little bit sick, right? And then the cold kind of, or if it's a family, right? The kid's sick and then one parent gets sick, the other parent and the other child, and it kind of like goes around the house. The virus is still there, but you're still making immunoglobulins against it and you still have those for a month and a half. Right. So the next time you accidentally like, oh, they forgot to eat the rest of their grilled cheese sandwich and that you eat it, right? You're not going to get as sick that time because you still have those IgGs floating around. The only one to cross the placenta, so it can protect the fetus. It fixes complement. It has that phagocytes have that FC receptor for IgG. So it does like everything. It's like super antibody. This is my personal favorite, IgA. It's a dimer, so it can bind 4. It's like 2 baby IgGs stuck together with their tails next to each other. So it has 4 antigen binding sites. It's for local protection. This says 13%. That's probably overestimating because you find it on mucosal surfaces. Mucosal cells make it. pretty much all over your body. 6 days for half-life, so it's not around as long, and it doesn't do any of those things. But it gives you local protection. For example, you have a lot of IgG in your mucus in that gut-associated lymphoid tissue, like right out of your stomach into the beginning of your small intestine, because you're getting exposed to all kinds of antigens there. And these IgA molecules are binding them, carting them around, taking them to the phagocytes, getting them broken apart so they don't make you sick. IgM is super huge. It's like 5 IgGs all stuck together in a ring. That gives it 10 antigen binding sites. So it's really good at this agglutination function, right? It's also kind of good at opsonization. It is always the first immunoglobulin that a plasma cell makes. All plasma cells, when they're first differentiated from the B cell, make IgM. And then after 1 to 2 days, they switch. We call it class switching. They switch to making one of the other immunoglobulins.
So that's the first, like, first response.
It's your first response, always. Half-life 5 days, so it's around for almost 2 weeks. It's too big to get across the placenta. It does fix complement, and it's always produced first. D, IgD is in dog always as surface marker for pre-B cell, for immature B cells. It's never in the serum by itself unless B cells are being killed, right? Or there's a overproduction of B cells for some reason. And IgE is also, look, very rarely, teeny tiny amount in the serum because it's attached to mast cells and basophils. So like IgD, it serves as a receptor for allergic reactions and parasitic worm infections, to trigger your immune system to fight those particular things. So it's not floating around in your serum either. So really when we say the antibodies switch from IgM to a different antibody, it's mostly G and A that they switch to. Okay. I say that is enough for today. Let me just stop that recording. Whew. Yes.